Hi, I'm Katie Danis, this is Beatrix the X chromosome, and welcome to Crash Course Genetics. Today we're looking at the story of a family, a rare genetic disorder, and one of the most pressing bioethical questions of the modern day. See, our friends Malcolm and Monica have 3 children, their 11-year-old daughter Lucy and 8-year-old twin boys George and Franklin. Lucy seemed like a pretty normal, healthy kid until the age of 3 when she began to show signs of intellectual disability. They took her for a genetic test and she tested positive for Fragile X syndrome.
Katie, what's Fragile X syndrome?
Slow your roll there, Beatrix. We're getting to it. Fragile X syndrome is a genetic condition that causes intellectual disabilities, behavioral symptoms such as speech delays and hyperactivity, and physical characteristics such as large ears, a prominent jaw, and flat feet. About 5% of females and 15% of males with Fragile X also have seizures, and due to the behavioral symptoms of Fragile X, some children who have it also have features of autism spectrum disorders and/or attention deficit disorder. Fragile X syndrome occurs in about 1 in 3,600 to 4,000 females and about 1 in 4,000 to 6,000 males. It is more common and tends to be more severe in males than in females because it is inherited in an X-linked dominant pattern.
Katie, what does all this genetic mumbo-jumbo have to do with the disorder being more common in males than in females?
I got you, Beatrix. Let's back up and define some stuff.
Here, educate yourself.
So we know that humans, like other eukaryotes, have DNA as our genetic material, the blueprint for our physical bodies. The building blocks of DNA are called nucleotides, a sort of computer code that tell our cells how to make different compounds.
I'm a coder, Jared, so I'm gonna code.
A gene is a series of nucleotides, and it is the basic unit of heredity. Some genes don't code for anything at all, but others code for gene products, proteins or RNA that fulfill functions in an organism. Taken together, these gene products can constitute traits, an organism's physical and behavioral characteristics. When a gene codes for a trait and that trait is observable, we say that gene is expressed in an organism's phenotype. For example, there is a gene in the DNA of pea plants that codes for what color the flowers of that plant will be, what its phenotype will be.
Katie, if all pea plants have this gene, why do some pea plants have white flowers and some have purple flowers?
Very astute, Beatrix. For the answer, we're gonna go to the thought bubble, which is definitely animated and not just me explaining concepts with kitchen items. We're on a budget. So we remember how Darwin explained biodiversity, aka the variety of life on Earth, with the theory of evolution. You start with one organism, and then it has offspring, and then those offspring have offspring, and so on and so on. And at any point on this tree, an individual may be slightly different than the individuals that came before it. Sometimes the individual will be able to pass down the trait to its offspring, and if the trait keeps getting passed down, the tree can end up looking very different. That's how we get variation among and between species, and how we know that this Clementine and I have a common ancestor. But hold on, you say! How can the offspring of an organism be different than the generations that came before it? The answer is mutation. As Professor Charles Xavier of X-Men points out, mutation is the key to evolution. A mutation is a change in the nucleotide sequence of an organism's genome, its complete set of genetic material. And in eukaryotes, like these oranges, this Clementine, and me, that's DNA. Sections of genes can get inserted, deleted, and transposed in all kinds of wacky ways. This can happen due to errors in DNA replication, cell division like mitosis or meiosis, or even other factors like exposure to UV light. Sometimes the mutation can make its way into the organism's gametes or sex cells during meiosis, which is how it gets passed down through generations. A mutation leads to a variant form of a gene, which we call an allele. Here's an analogy. You ask me for a cookie, and I say, which kind do you want? This peanut butter cookie, or this double chocolate chunk. Both of them are cookies, but they're each variants on a cookie. This is the peanut butter allele for cookie. Back to you, Katie. Thanks, Thought Bubble. Like there are many kinds of cookies in the world, there can be 2 or more alleles for a given gene. However, a chromosomally typical person has 2 alleles for each gene, inheriting one from their mother via egg and the other from their father via sperm. And these 2 alleles have a relationship to one another. Let's return to the pea plants as an example. This pea plant has 1 copy of the allele for purple flowers, big P, and 1 copy of the allele for white flowers, small p, yet its flowers are purple.
How did this happen?
This is because the purple allele is dominant to the white allele, and the white allele is recessive to the purple allele. Therefore, whenever the purple allele is present, it will mask expression of the white allele, so the flowers appear purple. It's not always so cut and dry, and it gets more complicated when you have traits controlled by multiple alleles and even multiple genes. Then you run into funky territory like codominance, incomplete dominance, and epistasis. I'll put links in the description for more information about those. But right now, for the purposes of this video, we just need to know dominant and recessive. For an example of a dominant heritable disorder, let's look at Huntington's disease. It's a rare neurological disorder caused by a mutant variation of the HTT gene on chromosome 4. If a female has one copy of the mutant allele and one copy of the normal or wild type allele, they will have Huntington's because the mutant allele is dominant over the wild type allele. Let's say this person has a child with a male who is unaffected by Huntington's. The male must have 2 copies of the wild type recessive allele. We call this homozygous recessive. And he has a child with a female who has 1 copy of each allele. We call this heterozygous. Their child gets 1 allele from each parent, so what are the chances the kiddo will have Huntington's? Well, we know they're getting a recessive wild type allele from their father. There's a 50% chance their mother will give them a recessive wild type allele, in which case the kid will not have Huntington's, and a 50% chance the mother will give them the dominant mutant allele, in which case the kid will have Huntington's. In that regard, Fragile X is similar to Huntington's, in that both are caused by a dominant allele. However, Fragile X is inherited a little differently from Huntington's, and here's why.
Are you finished, Mr. Slade?
No, I'm just getting warmed up.
Chromosomes are condensed versions of DNA. A chromosomally typical human has 46 chromosomes. 44 of these are autosomes, non-sex chromosomes that code for all kinds of things. 2 of them are sex chromosomes, whose primary function is to determine the biological sex of the person in question. Typically, females have 2 X chromosomes, and males have 1 X and 1 Y chromosome. There are exceptions and variations like Klinefelter and Turner syndrome that we unfortunately don't have time to get into right now, so for our purposes we'll just say that XX is female and XY is male. Huntington's is called an autosomal dominant disorder because its gene is located on chromosome 4, an autosome. By contrast, the gene that codes for Fragile X is located on the X chromosome, so we say that the disorder is X-linked. The reason it's more common in males than in females is because males have 1 X and 1 Y. So if a cisgender man gets a Y from his father and an affected X from his mother, that means that 100% of his X chromosomes will have the Fragile X mutation. By contrast, if a cisgender woman gets a normal X from her father and an affected X from her mother, only 50% of her X chromosomes will have the Fragile X mutation. To get into more detail, Fragile X is caused by a mutation in the FMR1 gene on the X chromosome. There's this repeating sequence of 3 nucleotides on the gene. Most people with 2 copies of the normal recessive wild type allele, wow, that's a lot to say, have fewer than 55 repeats of the sequence. People with the full Fragile X mutation have over 200 repeats. So that gives us this intermediate territory of people who aren't fully normal but don't have the full Fragile X mutation. So we say that people who have between 55 and 200 repeats of this sequence have a premutation. People with premutations can have their own phenotype that's not quite Fragile X and not quite normal, so for this reason they're normally not identified until they have a kid with the full Fragile X mutation. For this reason, people with the permutation are called carriers. They carry a copy of the affected allele and can pass it on to their offspring, but the allele isn't fully expressed in their phenotype. Going back to Malcolm and Monica, we know that Malcolm doesn't have the affected allele because he only has 1 X chromosome and he has a normal phenotype. So for Lucy to have the Fragile X phenotype, she would have to get the allele from her mother. That means that Monica has to be a carrier. She has the premutation.
Katie! If Monica and Lucy both have 2 X chromosomes, how come Monica's a carrier but Lucy has the full mutation?
Good question, Beatrix. That's because of 2 things called dosage compensation and X inactivation. Chromosomally typical females have twice as many X chromosome genes as chromosomally typical males. Some animals, including humans, solve this problem through dosage compensation, adjusting how many of the genes on the X chromosomes are expressed so males and females have equal X chromosome dosage. We achieve this through X inactivation in females by silencing 1 X chromosome at random and primarily expressing the genes on the other X chromosome. So it's likely that Monica's affected X chromosome was inactivated, while Lucy's unaffected X chromosome was inactivated. That explains why Lucy expresses the full Fragile X mutation while her mother Monica does not. So, now we understand how Monica passed her affected X chromosome to Lucy and how Lucy developed Fragile X. But how do we test for Fragile X? How do we know if someone has the full mutation or is a carrier? To help us understand, let's head to the thought bubble for a brief skit by our puppet acting troupe.
Hello, Doctor. My daughter Lucy and I would like to be tested for Fragile X syndrome.
Sure thing. We'll need to do a blood test for each of you, and then we'll use one of two possible lab tests to determine whether you have Fragile X. Come, let me introduce you to our lab tests. I am the Southern Blot Analysis Test. I separate DNA fragments via electrophoresis to detect the DNA sequence on the FMR1 gene. I can determine whether the gene has a full Fragile X mutation. And I am the Polymerase Chain Reaction Analysis Test, but my friends call me PCR. I make millions to billions of copies of your DNA, so the lab people can study it in detail. So they can actually use me to detect how many repeats of the CGG sequence you have. Therefore, I can tell you whether you have the pre-mutation. And did I mention I'm less expensive? Wow, that's quite the team you got there, Doc. Lucy, what do you think we should do?
I think we should go with PCR.
PCR it is. Yay! And scene.
Thanks, puppet acting troupe. So, Monica, George, Lucy, and Franklin all got tested for Fragile X via PCR. The results showed that Monica was a carrier. She had the pre-mutation, but Lucy, George, and Franklin all had the full mutation. Monica was devastated when she found out she was a carrier. She felt responsible for passing the affected allele to her children, but how could she have known she had it? She didn't get tested in infancy.
Katie, if we have the technology, why don't we just test everyone for every major genetic disease before they have kids and or screen them at birth?
That's an important question, Beatrix, and it's where we get into the ethical component of this whole shindig. The question of whether or not we should test people for genetic conditions before they have kids, and if so, to what degree we should test them, is a huge topic that we do not have the time to cover right now. I mean, genetic counseling is an industry that exists, and it is the subject of great ethical debate. So this kind of screening is happening and is being fought over, but we're not going to enter that discourse right now because it is too broad. Instead, we, Beatrix, are going to focus on the question of screening people at birth. Here's the big question. Is preventive genetic screening economical for society? Or is it more economical for society to bear the cost of caring for children with such severe disabilities? I argue, yes. And yes. Babies born in U.S. hospitals are screened for some rare genetic, metabolic, and hormone-related conditions at birth. The doctor pricks the kiddo's heel, takes some blood, runs it through a lab, and the results are ready within 4 to 7 days. The conditions newborns get tested for at birth vary from state to state. In my home state of North Carolina, newborns are screened for a number of conditions, including cystic fibrosis, sickle cell disease, and phenylketonuria. It can be helpful to know if someone has one or more of these conditions early on, because the person's family can then take preventive measures to help them manage their symptoms later in life. For example, phenylketonuria, PKU, is a metabolic disorder that can cause, among other things, brain damage and seizures. So if left untreated, it can impact a person's quality of life. As of now, there's no quick fix for PKU, but a person can manage their symptoms by following a diet low in the amino acid phenylalanine. In 1961, physician and microbiologist Robert Guthrie suggested screening for PKU at birth, so parents of kids with PKU could start them on a restrictive diet early on and hopefully help them decrease their symptoms later in life. We screen people for PKU at birth to this day, leading people who have the disease to receive early intervention and a better quality of life than that which they otherwise might have had if their condition hadn't been identified early on. We could do the same thing with Fragile X, screen people at birth so they could receive proper therapy early on and potentially have a better quality of life. However, there are two main criticisms of this approach. The first is economic. Fragile X is relatively rare. It occurs in less than 0.1% of the population. So it's reasonable to think that testing every single person for a disease that afflicts so few people is a poor use of resources. The problem with this criticism is that it ignores the potential payoff of preventive services. Preventive treatment and early testing can help a family avoid steep healthcare costs later in life. So it's a shame that preventive medicine is woefully underfunded in the US. Only 3% of our healthcare costs go towards it. Estimates vary on how much money, if any, we could save by prioritizing preventive testing and care. Some economists argue that it would save no money and would actually harm our healthcare system. But there isn't a definite definite answer to this question, at least not right now. I argue that the most important criticisms of screening newborns are bioethical criticisms, and there are a host. We are going to hit the highlights. Some critics of newborn screening point to the therapeutic gap, a phenomenon that occurs when screening technology surpasses treatment technology. By conducting all of these tests, doctors get inundated with information, much of which is not useful to them. Kind of how I felt while doing research for this video. The therapeutic gap can occur when we can screen for conditions before effective treatments become available, i.e. we have all this info and no idea what to do with it. This can lead to unnecessary anxiety for families and frustration for providers who may want to help but don't have a lot to work with. Another ethical challenge of preventive screening is its potential to exacerbate discrimination in healthcare and insurance. Let's take sickle cell anemia, an inherited condition that causes red blood cells to become misshapen and break down. It can occur in people of any race and ethnicity, but it's most common in people of African descent. Because we live in a racialized and racist society, people with sickle cell are disproportionately affected by medical racism. As a 2012 article in the Annual Review of Genomics and Human Genetics points out, quote, "The sickle cell screening history is littered with examples of racism and misinformation that resulted in employment and health insurance discrimination," unquote. In these cases, getting screened for a condition can be harmful to you because it puts you at greater risk of discrimination and requires you to run into racist barriers in the healthcare system over and over again. It's unjust, unethical, and downright infuriating, and we won't improve our preventive care until we address and dismantle these systemic barriers. So, would preventive testing for Fragile X be economical for society? Maybe yes, maybe no. It may have saved Malcolm and Monica some healthcare and testing costs down the road if their kids' conditions had been caught early. But asking whether it would be economical for society doesn't have a conclusive answer and is kind of besides the point. If we are truly dedicated to preventive medicine, we have to address socioeconomic barriers to healthcare. Otherwise, there's little point to testing people beyond collecting data for data's sake, which can be useful per se, but we have to keep in mind that those data may not serve human interests until effective treatments are made accessible. From a humanistic perspective, and medicine is a humanistic discipline, genetic testing can be useful when it helps people like Monica, Malcolm, Lucy, George, and Franklin manage their health, family life, and medical debt. And like a nucleotide in a DNA sequence, we view genetic testing not as a standalone concept, but as part of a whole system. That's a wrap on this episode of Crash Course Genetics. Beatrix, do you want to say anything in closing?
We chromosomes aren't much for talking, so I'm gonna cue you good folks in on the day in the life of an X chromosome in the best way I know how, with a musical number. Oh, puppet acting troupe, if you'll join me on the theater stage.